Okay, so hello everybody. My name is Mariana Varna and I'm Associate Professor at the Institut Galien at Paris-Saclay University. And in next few minutes, I will show you a few results about new nanoparticle formulation for cyclosporin and all assessment we perform in vitro. First of all, I will show you some information about Cyclosporin A, the structure, the origin, the mechanism of action. And after that, why Cyclosporin A is important in cardiac ischemia and reperfusion. And after that, I will show you our work obtained with Cyclosporin. This nanoformulation, I will show you how we obtain this new nanoformulation, and I will finish with some conclusion. So Cyclosporin A is a cyclic neutral lipophilic endocapeptide and has a 7 N-methylated peptide bond and can be synthesized chemically or can be obtained naturally by purification from different species of Tolypocladium inflatum, Aspergillus, Tolypocladium niveum, and other species. Cyclosporin A is already used in clinical practice. For example, we have restasis for ophthalmic emulsion, which is an ophthalmic emulsion, neural, which is a microemulsion, and Sandimune, which is an oil in water oral emulsion. And at the beginning, it was used to prevent rejection in organ transplantation, bone marrow transplantation, and more recently, it was used to treat inflammatory diseases like rheumatoid or psoriasis diseases. The mechanism of action of Cyclosporin A is now very well understood, and thus Cyclosporin A can penetrate cell membrane and can link to cyclophilin to form a complex, and this complex will inhibit calcineurin and finally will inhibit overexpression of a pro-inflammatory cytokine like interleukin-2 or interleukin-4. Another mechanism is by binding to nuclear factor kappa B activation, will inhibit its activation, and finally will inhibit the expression of pro-inflammatory gene. And a third mechanism is by binding to MPTP pore, that means a mitochondrial pore and transition, mitochondrial permeability and transition pore opening into the mitochondria. And this mechanism is very interesting for us. And it was applied for cardiac ischemia and reperfusion. First of all, what's happened during ischemia and during reperfusion? During ischemia into the heart, the cardiomyocyte will enter into anaerobic respiration, and this leads to a lower pH. The lower pH will block the opening of MPTP here on mitochondria. But when reperfusion is obtained, fortunately, this leads to a new secondary effects on cells, because the lactic acid is washed out, and this will restore the pH into the cells. And the restoration of the pH will lead to the opening of MPTP. And in this situation, the mitochondria functions is abnormal, and the opening of MPTP will allow the release of cytochrome C into the cytoplasm, and after that the cell will induce to the cell death. That's why inhibiting the opening of this MPTP should be a good strategy. And this was tested in 2008 by Piotr and co-workers. They administered into patients with cardiac ischemia and reperfusion Cyclosporin A, and they obtained a very interesting results. Later, in 2010, these beneficial results were also obtained in a followed-up study. But later, these results were more mitigated when they were conducted in larger randomized and double-blinded multi-center trials. The results obtained were explained by the fact, these contradictory results were explained by the fact that the cyclosporin was well-body biodistributed and finally led to a very low accumulation into diseased area. And in order to overcome this limitation, few nanomedicine were developed, and here you can see only a few examples of this kind of nanomedicine. For parenteral administration, the researcher obtained different kind of polymer linked to encapsulating Cyclosporin A with or without polyethylene glycol, etc., for oral administration and for ocular administration. In general, this nanoformulation increased the solubility of cyclosporin and show low toxicity, which is a good point. But they observed also a low entrapment of cyclosporin into this nanomedicine, and this could be a limitation. And in our laboratory, we have developed a new technique named Squalenoillation. That means we use squalene, which is a lipid, a natural lipid, biocompatible and biodegradable. And this lipid can be linked to a variety of active principles. And here you can see only a few examples for vitamin C, for adenosine, penicillin, acRNA. It's more difficult to link the acRNA, gemcitabine, and you can see we obtain every time we obtain nanoparticle. And we applied this technology to obtain a bioconjugate cyclosporin linked to squalene. And here you can see the chemistry. So you have here the cyclosporin, okay? Cyclosporin react with chloroacetic anhydride. And after that, we react with trisnorosqualenic acid, and we obtain this bioconjugate. So I am not chemist, but my colleagues who are chemists have the experience to do this steps. It was very difficult at the beginning, but now the technique is well mastered in our team. So we obtain this bioconjugate, as you can see here, squalene linked to cyclosporin. And after that, we dissolve this cyclosporin squalene in absolute ethanol at 6 milligrams per milliliters, and we added dropwise under vigorous stirring in a solution of dextrose 5%. Why dextrose 5%? Because it's an isotonic solution. We wish administered this new nanoformulation into blood through intravenous administration. After that, this suspension is evaporated by using a rotavapor in order to completely evaporate ethanol, and we verify that the ethanol is completely evaporated. And the final solution obtained at 2 milligrams per milliliters was analyzed by using a classical machine, as you can see here, a zeta sizer. And we can evaluate the mean diameter, the polydispersity, and index and surface charge. And our results, you can see here only one example, show a mean diameter of 170 nanometers, a very good polydispersity index, and a zeta potential of approximately minus 14. Moreover, we evaluate the size of this nanoparticle by using a cryogenic transmission electronic microscopy, which is a gold standard to be sure that your nanoparticle have the good shape and to measure also the size. And we applied cryogenic transmission electron microscopy because it is more adapted to this lipid kind of nanoparticle. Our image show a very nice spherical image, as you can see here, with a mean diameter of 105 nanometers. And after that, we evaluate the stability of nanoparticle, and we evaluate the stability at 2 concentration, 2 milligram per milliliters and 4 milligram per milliliters, and 2 condition, room temperature and 4 degrees. And we evaluate this by using DLS, Z and zeta sizer. We evaluate up to 30 days. And as you can see here, our nanoparticle, squalene, cyclosporin, were more stable at 2 concentration when they are kept at room temperature. But at 4 degrees, they will precipitate. So this information was very important for us because we know that in this situation, for this nanoparticle with this active principle, we should keep our nanoparticle at room temperature and not at 4 degrees. Another example, squalene adenosine, we have to keep at 4 degrees, but not at room temperature, because at room temperature they are not stable. So this information was very important for us. Okay, further, we evaluate the toxicity of our nanoparticle to be sure that they are not toxic, they don't destroy our cells. And here you can see 2 concentration. We tested 2 concentration, 12 and 16 microgram per milliliters. And you can see here, in general, they are not toxic. So we use 2 saline mouse cardiac endothelial cells and rat cardiomyoblast, and this kind of saline are generally used for this kind of techniques. Endothelial cells, because they uptake, they are the first line in contact with nanoparticle, and the cardiomyoblast are cardiac cells, so they represent our target. So further, we evaluate the uptake of this nanoparticle by the cells, and we assessed the uptake at 2 concentration, 12 and 16 microgram per milliliters, and up to 24 hours. And here you can see the results obtained. In blue is nucleus colored with DAPI. In green, you can see phalloidin. So phalloidin colors microtubule and microfilament. And in red, we obtain the nanoparticle, and with body P inside. Body P is a lipidic color, is a lipidic fluorescent, and we include body P inside the nanoparticle when we prepare the nanoparticle, when we obtain the nanoprecipitation. That's why body P is inside our nanoparticle. And you can see here that for this kind of cell line, cardiomyoblast, we can see a very low uptake we can detect a very low signal when the nanoparticles are not very concentrated, but this signal is more important when the nanoparticles are more concentrated, and as you can see here at 18 hours. In parallel, we perform also the control with only body P, so no nanoparticle, but only body P. And you can see that no fluorescence, no red signal detected at 24 hours. And we also evaluate the uptake on cardiac endothelial cell lines. And as you can see here, for both concentration, we detect very early uptake. So the cells uptake in a high amount this nanoparticle. And in parallel, you can see here only the body P, so the control, which is completely negative. And the difference between these 2 kinds of cells was explained by the fact that endothelial cells possess uptake capacity which is not a characteristic for cardiomyoblast. That's why we test this. We have tested these two kinds of cells. And finally, we evaluate the cardioprotection of this nanoparticle. And to evaluate this cardioprotection, we plated cells into a plate for 24 hours. After that, we rinsed and we applied the treatment with nanoparticle or with free nanoparticle containing a cyclosporin, or with free cyclosporin, or with a squalene, only squalene nanoparticle. And we applied this treatment for 24 hours because we observed a very high accumulation at 24 hours. And after that, we rinsed and we changed the medium with another medium specific for hypoxic condition. That means without glucose and without fetal bovine serum. And as you can see here for these three columns, the first plate was kept only in hypoxia for 6 hours. Another plate was kept on hypoxia for 6 hours, followed by a reoxygenation for 30 minutes. And the third one was kept on hypoxia for 6 hours, followed by a reoxygenation for 1 hour. And we analyzed the results by using two different tests, MTT, which is a mitochondrial test, and LDH assay, which indicate the cell membrane integrity. So for MTT, when the bar are high, that means we have a good level of cell survival. And for LDH assay, when the bar here, as you can see here, are low, that means a very good survival because the cell membrane is intact. And as you can see for this first kind of a series of experiment, when the cells are incubated with nanoparticle squalene Cyclosporin A, they are protected from cell death, either in hypoxia or hypoxia followed by reoxygenation, or hypoxia followed by reoxygenation 1 hour. So this protects the cells. And these results were also confirmed with LDH assay. Every time when the cells were incubated with nanoparticle, they were more protected. And this was the case for H9C2 cell line. We also tested endothelial cell line, and as you can see here, the results were more mitigated. And the explanation we did for this is that these cells uptake a very high amount of nanoparticle, and maybe it's a little bit toxic for cells. So maybe for endothelial cells, we have to apply a less amount of nanoparticle. But anyway, they were protective, also protective for this cell, for these cells. So in conclusion, we obtained this new type of nanoparticle based on the covalent bond between squalene and cyclosporin A, which is a new strategy, is an innovative strategy. The bioconjugation forms nanoparticle of controlled size in dextrose solution. Nanoparticle obtained were stable at room temperature for several weeks. A strong protective effect on cardiomyoblasts and the more mitigated one on endothelial cells was observed, and deeper works are ongoing in order to understand the exact mechanism of protection. That means how cyclosporin can act when is uptake as nanoparticle and when is into the cells. How the link between squalene and cyclosporin is cut in order to obtain only cyclosporin, which is an active molecule. So a deeper work, we are doing now a deeper work in order to understand this. And before ending, I wish acknowledge a lot of people who work on this project, and of course the different funding financement. I wish to inform you that we have now a special issue, and I am guest editor for Pharmaceutics, and the deadline is 1st of September for this year. So if you have a paper on this field, you can submit it. So thank you very much for your attention, and if you have any questions,